We culturally imagine cognitive enhancement as a simple, infinite trajectory, a chemical ceiling that rises as long as the dosage does. But biological systems operate on a strict logic of balance. When a synapse is flooded with synthetic neurotransmitters, the brain initiates counter-regulatory measures to preserve its original equilibrium. The formal study of these substances began in the 1970s with Romanian psychologist Corneliu E. Giurgea. He coined the term nootropic, combining the Greek nous for mind and trop for turning. Giurgea set a high bar for this classification. To be a true nootropic, a substance had to enhance learning and memory while lacking the psychological and pharmacological side effects common to other psychotropic drugs. Modern usage has fragmented this definition. Today, mild neuroprotectants and botanical supplements are grouped under the same label as aggressive pharmaceutical stimulants. This botanical class includes substances like ashwagandha, turmeric, and ginseng, herbs with long histories in alternative medicine, now processed into modern capsules. At the other end of the spectrum are hardcore psychostimulants. These are regulated medications, like Ritalin, synthesized to treat diagnosed conditions like ADHD and narcolepsy. While these molecules definitively alter executive function, their efficacy is not universal. It is strictly conditional upon the baseline neural architecture of the person taking them. This creates a fundamental boundary. The same chemical that repairs a cognitive deficit in one brain may produce a very different result in a brain that is already healthy. To understand this boundary, we have to look at dopamine. In the prefrontal cortex, this neurotransmitter is the engine for working memory and cognitive control. This pathway is the primary target for mixed amphetamine salts and methylphenidate, stimulants often used off-label by healthy individuals seeking a performance edge. Mixed amphetamine salts increase chemical availability by blocking the dopamine transporter and reversing its action, forcing dopamine and norepinephrine back into the synapse. These salts further heighten the effect by inhibiting the enzymes responsible for breaking those neurotransmitters down. This process results in an accumulation of neurotransmitters that saturates the receptors within the synaptic cleft, forcing a change in neural signaling. This chart shows the inverted U relationship between dopamine and cognitive performance. Peak mental function occurs within a narrow range. An ADHD brain sits on the far left slope, below the functional threshold. Stimulants bridge this gap, pushing their performance up the curve to the optimal apex. However, a healthy brain starts near that optimal peak. When it adds a stimulant, it moves past the peak and down the right-hand slope. The chemical surplus overwhelms the system, and cognitive performance drops. This explains why oversaturating a healthy prefrontal cortex often replaces executive focus with impulsivity, restlessness, and clinical anxiety. While stimulants target dopamine, memory consolidation relies on the cholinergic system. In the hippocampus, signaling depends on a steady supply of acetylcholine. Substances like donepezil and huperzine A act as inhibitors, preventing the enzymes in our brain from breaking that acetylcholine down. This brings us to Piracetam, the prototype drug Giurgea used to define the entire nootropic field. At the molecular level, Piracetam modulates AMPA receptors, which are essential for long-term potentiation, the physical process that allows the brain to store new information. But there is also a physical hypothesis for how it works. Piracetam appears to partition itself into the fatty bilayer that forms our cell membranes. In aging brains, these membranes lose their fluidity. They become stiff, which slows down the movement of receptors and the release of neurotransmitters. When the drug integrates into the membrane, it restores that fluidity, allowing signals to pass through the cell more efficiently. This mechanism was put to the test in a study by Mueller and colleagues, who compared the drug's effect on aged brain tissue versus young, healthy tissue. The results showed that while Piracetam significantly improved fluidity in damaged or aged membranes, it had zero physical effect on young, healthy brains. Because healthy tissue already maintains optimal membrane fluidity, there was no structural deficit for the drug to repair, and volunteers showed no measurable cognitive gain. This reveals the structural limitation of these compounds. Their effectiveness is dependent on an existing biological deficit. Pushing a homeostatic system has long-term consequences. To defend against a chemical flood, the brain begins a process of downregulation. To prevent overstimulation, receptors actually internalize, receding into the cell membrane. This creates a physical dependence just to maintain the original baseline. Beyond the biology, there's the market itself. A surveillance study found that 68% of commercial nootropic products originated from illegal supply chains. More concerningly, 50% of the supplements tested, many marketed as natural, contained unapproved research chemicals like Nuapept and Phenibut. In the search for a pharmacological shortcut, healthy individuals are often bypassing regulatory safety nets and exposing themselves to unregulated cardiovascular risks. When we step back, we can see three very different trajectories for cognitive intervention. In the first panel, an Alzheimer's patient's declining baseline stabilizes. In the second, an ADHD patient reaches a functional peak. But for the healthy user, a brief subjective spike is followed by a steep drop as receptors burn out. These results confirm that neuropharmacology does not scale intelligence, it corrects specific pathway malfunctions. The focus healthy users often report is typically the result of the placebo effect, combined with increased physiological arousal, essentially mistaking a racing heart for a sharper mind. For those with a healthy baseline, sustainable enhancement is not found in a pill, but in the biological systems that drive neuroplasticity. This means prioritizing the only core pillars scientifically proven to work: sleep architecture, aerobic exercise, and metabolic health. Cognitive power is a function of homeostasis, not its disruption. To truly optimize the mind, we must